Hi, everyone. I'm very pleased to welcome you to INDICAL's webinar on avian influenza, sharing experiences between Southeast Asia and Europe. My name is Norhan El-Hakim, and I'm Head of Marketing and Communications at INDICAL. The following webinar was recorded on November 30th and moderated by APAC Director of Sales at INDICAL, Alison Digney. We hope you find the session insightful.
So what you can see is today's agenda. We're going to have a very short introduction on INDICAL, veterinary diagnostic company. Then we will have two presentations, one from Jean-Luc Guerin and a second one from Erik Karlsson, followed by a question and answers and some closing remarks. Once again, please add any questions at any time into the question pane as we go through. So just a quick introduction into INDICAL. So INDICAL is a dedicated veterinary diagnostic company that is here to improve diagnostics for better animal health and its impact on human health. INDICAL has some cutting-edge technologies and continues to innovate to improve how diagnostics can be applied for the management of diseases. So our priority as a company is to empower customers with fast, reliable solutions that ensure accurate and actionable results. Really, what we need to do in the world, whilst we're having increased outbreaks of disease, is give solutions that give vets and governments the ability to not just react, but be proactive in monitoring and preventing the spread of disease. And we are working with industry leaders to ensure that we have a complete toolbox for you. We want to work with you and learn from you so that we can provide a safer, cleaner world. Next. So our solutions aren't just regular diagnostic kits. We're looking to take the next step in diagnostics to give you actionable solutions, innovative solutions. We have a 25-year history, which means that we've got quality, reliability, and the ability to take everything from a sample through to an actionable result, helping you monitor and react to disease. Next slide. When we look at poultry is one of the leading protein sources in the world, and we have a growing capacity for the diagnostics in poultry. We have what is routine diagnostics using qPCR, and these cover a range of different things, including influenza. Influenza, we have a screening tool which allows you to accurately and fast detect influenza A in multi-species. And then more importantly, the ability to look at high pathogenic strains, including H5, H7, H9 in a triplex assay with a control. We also have ELISAs, which is critical when we move forward in looking at how vaccines may be used in managing disease as the prevalence of avian influenza increases globally. We also have some newer technologies, such as our air sampling technology, which can be used for routine monitoring of flocks, but also for surveillance work. And we also have the ability to do field diagnostics with the INDIFIELD, which is a 9-channel qPCR instrument that runs off a battery that can be taken into the field to give you accurate, fast diagnostic solutions. Coupled with our complete workflow solutions for molecular biology with sample preparation and also qPCR reagents. Next, please. So we're a company that is short by name but long in history. With over 25 years experience, we started as a veterinary service laboratory and have evolved over many years. And today we become one of the forerunners in new technologies for monitoring and managing disease in animals. So I would now like to present our first presenter, Jean-Luc Guerin, who is a doctor of veterinary medicine, has a PhD, and is serving as a full professor in poultry medicine at the National Veterinary College of Toulouse in France. He's diplomat on the European College of Poultry Veterinary Science and director of the residency program. Jean-Luc is head of Host Pathogen Interactions Joint Research Group, covering virology, prions, bacteriology, immunology, and epidemiology of infectious disease in animals. In a One Health perspective, his own research is focused on viruses of poultry and for some years, pathobiology of highly pathogenic avian influenza. A priority is the development of innovative approaches to viral detection, including from environmental and wild birds poultry interface. Jean-Luc.
Thanks. Many thanks for the kind introduction and for the invitation to share some information and I would say some ideas regarding the current situation of avian influenza in Europe. And obviously, I think that my talk will be very greatly completed by the talk of Erik later on. So a short feedback and some ideas regarding the challenges for the control of avian influenza. As we know, the situation since at least 1 year, but for some years now, is very difficult due to one virus, which is linked to the Guangdong 96 lineage. And there is a specific clade, namely 2344B, which is circulating in Western Europe and now in United States for years. And one specific strain, H5N1, is now challenging the poultry industry. The circulation is widespread, and in this panel, you see the report of cases during the last season, I mean until August. The green dots refer to the cases in white birds, and the bluish dots refer to the cases in poultry farms, more or less. The key informations here is that you have obviously a very large amount of cases along the Atlantic coast, and that you have a significant number of outbreaks in specific regions in Europe. And I will focus on France because I'm French, basically, and we have a long story with avian influenza, but we had a lot of cases last winter in northern Italy, and in this specific situation, the key poultry species involved was obviously turkeys. You will see that in France, the key species is duck. I would say the different strains of duck. And we have different situations in different countries. But basically, the story is that you have a circulation of virus in the wild bird sector and introduction in different regions in Europe. That was the situation until August. And since September, we have still a large amount of cases, largely on the Atlantic coast, and it's more or less the same story in United States. And on the left panel, you see that in UK, the situation is very bad also. Since October, they had more or less more than 200 cases, and with let's say a concentration of outbreaks in this region here. So what we can say now is that contrary to the situations in the previous years, we observe that we did not observe any stop during the summer season. And that we think that that means that at least in the white bird sector, we could have more or less an endemic circulation of virus, regardless of migratory birds. And we observe sporadic introductions in the poultry sector in different regions of the western part of Europe. If I focus on France, the situation on the last season, I mean winter, spring this year, was, as explained, two main regions concerned, and these regions were considered as high risk of introduction of avian influenza and of diffusion of avian influenza. And the key question is, why do we observe this kind of concentration? What are the risk factors? And we try to investigate this for some years now in my group to try to clarify which is the key risk factor and which is the key factor to control the diffusion of virus. And we observe this year two main waves of introduction. The first one in the winter season in the southwest, which is the main traditional duck production region, and the second one in Pays de la Loire and Brittany. And it was linked to a later wave of cases in spring, I mean in March, April. As you see in the right panel, a lot of species were involved, including obviously ducks, but also gallinaceous birds. And we see that in some regions we have a mix of ducks and gallinaceous birds, and this mix in the same region is of high risk of diffusion of virus. So we try to investigate the cases on the field to try to identify the clinical picture, and to the lab to try to identify the best way to do detection of virus. Just very quickly, some pictures we observed on gallinaceous versus waterfall poultry. On gallinaceous birds, we observe traditional, I would say, classical pictures linked to vascular damages, necrosis of vessels, edema. In the same way, we observe here necrosis of vessels and an affection of the genital tract with egg drop syndromes and discoloration of eggshells. So it's classical. In breeders or layers, we observe this kind of picture, which is absolutely not specific, but in most of cases, it was almost the only lesion observed on the genital tract with hemorrhages and congestion of the ovaries. In ducks, we observe mostly, we used to observe sign, and it is very, let's say, traditional, classical in the Asiatic H5 viruses. And this is very typical, not really specific, but it is one of the most important first clinical signs observed in these birds. But here again, we observe some evolutions, and in the most recent cases, we observe that these type of clinical signs may be less evident, much milder than the cases observed last year. But we are still investigating this. The key picture in waterfall. So the key issue is what is the way of dispersion of virus, and we try to investigate this in different types of poultry species, and we did environmental sampling linked to classical official sampling, I mean swabbing. And what we observe is that at least in ducks and geese, we observe a very important shedding of virus through aerosols and through dust. And we compare the sensitivity of techniques of sampling, and what we observe, at least in ducks, is that environmental sampling, either aerosol sampling or dust sampling by wiping, was at least as efficient as the traditional swabbing and mostly tracheal swabbing, since the viral expression is much stronger, at least at the very beginning of infection in the oropharyngeal area. So one of the important message is that the environmental sampling may work properly for the detection of this virus, at least in ducks. And why do we observe this? Because before the onset of clinical signs, we know that we have a period of, let's say, up to 1 week or even more than 1 week of non-clinical shedding. That means that the ducks are shedding, excreting virus in large amounts without expressing any clinical sign. And this is very tricky. And that's why the, let's say, molecular surveillance may be very important in that stage, in this stage. And what we know is that the situation and the dynamic of infection may be very different if we compare the ducks and the gallinaceous birds. This period may be much shorter in gallinaceous birds such as turkeys and broilers. This is, in my opinion, a very important information. So we try to map the risk and at the country and the regional level. And to make a long story short, what we show, based on the very first outbreaks almost 5 years ago, is that on territory, the density of duck farms is a key risk factor due to the potential of dispersion of virus by this species. And we observe that this risk factor is very important, and the density of duck farms in France, in the French context, may play a significant role. And we propose a risk mapping. We published this 3 years ago, and this mapping was used to implement a new regulation in France with these yellowish zones that we call zones of high risk of diffusion, ZRD in French. And what we observe this year is that the last cases, as I showed you previously, in the southwest part and the western part of France, merge almost totally with the risk zones, the zones of high risk of diffusion. That means that actually the density of duck production is a key driver in the infection, at least in France. It's not the same story in other countries. We know that in Italy, the turkeys may be the driver and the situation may be different. So anyway, in the French context, we were able to suggest that the density of duck farms is a key driver. So to go to the end, how can we control this risk? The big picture of the introduction and diffusion of virus is now in Europe like this. We know that in a territory, in a region, the first introduction comes from wild birds. That does not mean that you will have droppings of these birds in the poultry farm. It's not so direct. You may have environmental contamination and then introduction of virus by the door, by failure in biosecurity, or by aerosol, many ways, and then diffusion from the index farms to other farms. And we know now that aerosol diffusion may be very important, and dust contamination may be important by people, by any mechanical vectors, and then transportation with animal, cages, material, and people may play also a role. And among the black boxes, we have one black box, regarding the role of the commensal birds in the dispersion from farm to farm. And we don't know, to be honest, if they play a significant role. We don't think that they are a key actor compared to aerosol, to biosecurity, but they play a role. And this is very important to clarify, since we know that at least in some European countries and even more in France, the outdoor poultry system is very important. So the key issue now is how can we still manage to raise poultry in this condition. And the investigation of a specific role of commensal birds will be very important in the future to adapt properly the control of introduction of avian influenza in this specific system. So we try to implement some regulation, and so far the only way is restriction. That means confinement of birds with protection of drinkers, feeders, and depending on the level of risk of introduction from the avifauna, we will adapt. And for example, now in France, in UK, and in several countries, we have considered at high risk. That means that there's a complete castration of all the birds. And it's not easy to implement this. And if we consider all the ways of the potential ways of introduction and dispersion of virus, this is huge. And we immediately understand that it will be very difficult to control this by classical biosecurity measures, since the risk of contamination is extremely high, and we think that the minimal infectious doses is very low, at least in very sensitive species such as ducks and turkeys. And the minimal infectious doses is critical to consider the way we can manage the protection of poultry farms. And we were surprised by these properties of dispersion, of contamination. And we know now that biosecurity is a key, but with limits. And we are very, it's very difficult now to control the dispersion of virus only by this way, to be honest. So the responses now are based on depopulation, including preventive culling, since we know that at least around cases in ducks, the potential of dispersion of virus is so high that we cannot manage to keep the birds alive in the neighborhood of this farm. So the preventive culling is almost mandatory, based and associated with surveillance. And we know that we have some issues even now, today, regarding the capacity for culling and disposal of carcasses. And the actors and first the vets have to do different things, make diagnostic, test, and even participate to culling operations. So it's it may be very difficult to manage to play all these different roles all together. So the next step could be and will probably be vaccination. And the objective of vaccination would be not only to block clinical signs, but first block viral circulation in duck parks. And that means that we absolutely need to be able to perform a diverse strategy. In France, we started focusing on ducks based on all what I explained. And we try to investigate the efficacy of vaccines on ducks, and trials are going on and on a good way now, with 2 vaccines included in the tests. And in these tests, we, of course, consider the efficacy, the protection against the challenge, but also the feasibility and the capacity to do dieback. That means to discriminate the response against the vaccine and the response against a wild type virus. So I would like to stress at this stage on the fact that even if we have a very good vaccine, we will only be able to use it if we implement first biosecurity and an intensive program of surveillance. And I think that we will talk about this later with the talk of Erik. Surveillance will be a key if we want to manage properly the vaccination program in the future. And I would like to stress also on the fact that we need a global vision, at least at the European scale, but at the, I would say, global scale, to accept this change in the strategy of protection, considering that we can implement vaccination and control the disease. That will be the key. And that means that surveillance in the wild bird sector and the poultry farm sector will be mandatory. Implementation of biosecurity will still be a priority, even more in the densely populated poultry areas, as we observe in France, in northern Italy, in some other regions in Europe. And the implementation of vaccination will be only, I would say, sustainable if we consider it closely together with biosecurity and surveillance. And with this, I would like to stop.
Thank you very much, Jean-Luc, for your presentation. I just want to remind everyone that we have the chat box. If you do have any questions, please put your questions in there. We will address them at the end of Erik's presentation. And now I'd like to introduce everyone to Dr. Erik Karlsson. Dr. Karlsson serves as the Deputy Head of the Virology Unit at the Institut Pasteur de Cambodge in Phnom Penh, Cambodia. He is a director of the National Influenza Center and a regional WHO H5 reference laboratory, and is the coordinator of the WHO COVID-19 Global Reference Laboratory. Dr. Karlsson is an internationally recognized expert in respiratory viruses, especially those with zoonotic and pandemic potential. His research focuses on field-forward technologies to improve surveillance and early warning as a One Health interface, especially in traditional food markets. He is also the founder of the Consortium of Animal Market Networks to assess risk of emerging infectious diseases through enhanced surveillance. Thank you, Erik.
Thank you very much, Alison. I hope you can hear me okay. I can see my screen as well. So good afternoon, everyone. As we said, my name is Dr. Erik Karlsson, and I am going to talk a little bit about avian influenza here in the Asia Pacific, mainly focused on Southeast Asia and specifically Cambodia, since that's where I'm based. But before I go on, of course, I want to thank all of the staff in the laboratories here, especially the scientists, the field teams, and the technicians that do a lot of this work and go out in the field, collect this data. So similar to Jean-Luc's presentation, we'll start out with a map. As you can see, obviously this is data from 2022, a number of AIV events reported globally. We had a nice overview from Jean-Luc about the situation in Europe. Of course, we do have situations in North America, now apparently moving down into South America and Africa. But really, we're going to focus here on the Asia Pacific region. As you can see, there's probably in your perception a lot fewer dots as compared to Europe and things like that. But it's likely that there's a lot of undetected or unreported AIV outbreaks or incidents here in the region. We have to remember that AIV has been endemic in Asia since the first outbreaks in Hong Kong in 1997, and then of course spreading throughout the region in 2003, 2004. Highly pathogenic avian influenza has been a major problem, but these viruses have probably been here for centuries, if not longer. In Cambodia specifically, avian influenza really is endemic. At any given time when we go out into the field, about 20 to 40% of chickens are going to be positive and about 30 to 50% of ducks, depending on the season. And so we've really been doing surveillance in live bird markets and other important hotspots in the country for over 10 years. And so if I take all of our human cases and our poultry outbreaks and years worth of data and plot those on the plot that you see here, you see some pretty definitive peaks and valleys. And what these actually correspond to is festival periods. And these festival periods are associated with increased poultry production, poultry movement, purchasing, and sharing of food. So, these outbreaks and waves of avian influenza that we see, irrespective of subtype, really have to do with social and cultural factors that can actually drive them forward. And that really circulates around the poultry value trade and things like that. We don't find as big of an association with wild bird migration or anything like that here in Cambodia. And it's more to do with the poultry trade around the region and within the greater Mekong subregion. So, we've been doing this for a long time, and the viral subtypes that we see here in Cambodia really do kind of reflect other things that are going on in the region. Obviously, we'll start with H5. Early in introduction here, it was clade 112, which some of you may be familiar with. We then had an introduction of 2321, and now we've moved on to 2344s, very similar to the rest of the region. Obviously, these viruses are causing major problems, not only in Europe, but are still causing problems in Asia and appear to have circled back around the globe and are now, we see H5N1, Eurasian lineage 2344s in Asia as well. They do cause mammalian infections, which are a big problem. And the bigger issue here in Asia is that they're also circulating with other things. So, we still have H7 viruses, which are a major problem. Obviously, everyone remembers Anhui lineage H7N9 viruses, but we now have these Jiangsu lineage H7NX viruses, which are reassorting like crazy here in Cambodia. These have been associated with human infections in the past. And then, of course, H9 viruses, which are endemic throughout the region, and including here in Cambodia. I think all of us that are aware of AIV know that the internal genes of H9 are quite important for transmissibility and circulation of avian influenza. They're likely circulating throughout the region, and even here in Cambodia, we've had 2 human cases of H9N2 in the past 2 years, one in February and one in March 2021 and 2022, respectively, both in Siem Reap province. And we'll talk a little bit more about those later. But there's also subtypes of concern that are circulating in the region, and we do detect these in Cambodia as well. So they're what I consider novel or understudied subtypes, so things like H6, H10, H11, avian H1s, avian H3s, and now we're even finding things like H14s in domestic poultry here in Cambodia, which is a big deal, as they were previously thought to only be associated with wild birds. So you can see that there's a major concern here that if the Eurasian 2344 H5N1 strains do get into the region and start taking over from the other subtypes, which they likely will, as we see, they could easily reassort and become a completely new strain as well. And that's really why we need early warning, and that's why surveillance is so important. So we need to get ahead of these diseases before they cause outbreaks or emerge into new populations. And so what's important for early warning really is to anticipate these viruses, to prevent them, so to actively reduce and protect against them, to be able to detect them, which is a major concern, obviously, and then obviously to be able to respond to them when they do emerge. And so the issue with early warning, it means it requires active surveillance, but But active surveillance, as some of you may know or are currently doing it, it's costly and it's time consuming, and it requires a lot of manpower and field work and laboratory work to maintain these systems. And oftentimes, you know, especially with new or emerging pathogens, it's often not in the easiest of places. So here you see people from my institute out in the field actually sampling wild birds. And so here in our unit and in collaboration with lots of people, we're really interested in improving surveillance for early warning. So there's two ways we can do this really. One is through sampling and one is through testing. And so here is your typical Cambodian live bird market. This is in the largest bird market in the country, Orasay market here in Phnom Penh. And as you can see, obviously we could go through and sample every single one of these birds that they are currently slaughtering. That would be a way to get a very good prevalence, but it would also take a lot of time, a lot of work. But we can take environmental samples, as I think you saw from Jean-Luc's presentation. There's lots of things that we can sample, and we've been sampling these for a long time. So things like surfaces, cages, wash water, feathers, and then as Jean-Luc mentioned, dust and aerosols are equally important. So I'm going to focus on some of the work we've been doing on aerosols. And so, you know, really, I think globally there's been a focus on how we can improve environmental sampling, especially with the COVID-19 pandemic. This has been shown to be very important for human surveillance, but we're also had many other studies have shown this is important for other respiratory pathogens such as AIV. And so we've been conducting aerosol sampling in live bird markets. So here you see an air sampler next to some ducks, and here you see it next to some next to a slaughtering area, and really looking at paired poultry and air samples over time. And what we find is that, you know, we can detect a number of different places where there's actually virus in the air. And so here I've just plotted, you know, detection in poultry on the left, and then as you move further away from the actual poultry themselves, and we can still detect it even up into food sales, which are physically separated by walls from the slaughtering areas. We also, in the birds, again, we're talking about these major subtypes, H5, H7, and H9. In the air, we are detecting the exact same subtypes. So we find that these are a viable option as per, you know, previous studies and what Jean-Luc has shown as a way to monitor the high-risk environment of things like live bird markets and farms. Another thing that we've been doing, and as Allison showed in the beginning, is using these handheld devices. So these are the AeroCollect samplers from INDICAL. Here you see we're using them with ducks, but also with in a slaughtering area. And you know, the quick question here is, and I don't have all the time to show you a bunch of data, but can we actually detect AIV in live bird markets using these handheld air samplers? So just in a quick experiment, we go in and sample, you know, chickens, ducks, these slaughtering areas, and even using, you know, these food prep areas as a control. We're running these for about 10 minutes each in each place, and then flushing the cell with about 30 microliters of fluid. And indeed, in this sample here that they're doing the defeathering, we actually found it was positive for H9N2 that day. This was not performed during the peak of AIV, but you can see on any given day, you can walk in and detect influenza in this situation. The other thing that we've been doing is using these personal wearable air samplers that can be set to actual breathing rates, and so, you know, using these air samplers, we've actually found that there's a 100% chance of an individual being exposed to a viable, so we're actually able to isolate these viruses out of these samplers, a viable H5N1 or a viable H9N2 virus in just 30 minutes of use during the peak AIV period. So that's during those peaks that I showed you earlier in the talk. We also want to use these in novel places. So I know this is a talk about avian influenza, but obviously the interfaces are quite mixed here. So things like using them in pig populations. So here you see us using these air samplers in pig sampling, and that work is ongoing. We are also using them to try to understand if we can pick up, say, spillover infections in wild and captive animals, which we have seen here in Cambodia in the past, but also for novel viruses, but also for things like bats. So can we actually use environmental samples in bat sampling instead of having to capture bats and things like that, which are relatively dangerous operations. So here you see an air sampler with a bunch of bats flying out. And we're even trying to use them remotely. So here you've seen we've put an air sampler on top of a drone, which we intend to fly into areas that maybe we can't reach, or other things that potentially we can try and pick up what's in the air in environments before we have to enter caves or other areas. The other thing we want to talk about today, and I'm sorry I don't have a lot of time, but is testing. So obviously we want to increase our ability to test these samples as well as to collect them. So testing and diagnostics are vitally important, not only to increase sensitivity and specificity, but also the versatility of what we're doing and to discover new high-risk pathogens. One viable option, as you saw, is multiplex testing. Obviously, being able to test environmental samples for multiple pathogens all at once is ultimately important because it both reduces our time to detection, but also decreases our staff time and potentially costs associated with that. We've done some work with this INDICAL virotype influenza A kit. Basically, the first question that we asked was, can it effectively detect all of the circulating strains and clades in the region here in Asia? And I can confidently say that yes, we were able to detect the majority of these strains here. So here you see listed what we've tested with this kit, and we are continuing to test this kit alongside other assays for usability in our surveillance systems. You're probably also asking yourself, if you're from a lab like mine that does both human-animal interface, what about humans? Obviously, things like our ILI, our influenza-like illness and SARI surveillance systems are really critical for seasonal influenza, as well as things like RSV and SARS-CoV-2, but they're also critically important for detecting spillover infections. So basically, we wanted to ask the question, can commercial human multiplex assays, so things that are meant to pick up human seasonal influenza, as well as RSV and SARS-CoV-2, do they have a potential for AIV infections? And so what we did was compare your standard assays, so things like the FAO recommended AIV detection protocol for the region, with as well as the CDC and VIDRL protocols that are used under the Global Influenza Surveillance and Response System, with 3 commercial kits. These are multiplex kits from several different companies. They all have different advantages and disadvantages. But the key here is that, as you can see at the bottom of this table, they were all able to accurately, within one log, pick up a number of different avian influenza strains, which is great for our surveillance systems in humans, because it means that we potentially won't miss any spillover infections as they come through our systems, which is a critical point in being able to detect these rare events. We're also trying out the pen-side solutions that were mentioned earlier. So we've got this in-the-field device in hand. We're continuing to validate this as well. And then, of course, sequencing. So in-field sequencing. We've done a lot of work with our colleagues to develop rapid AIV surveillance using integrated barcodes. And so what we've been able to do is work with our collaborators. I put the protocol down here at the bottom at Johns Hopkins, where we've developed integrated indexed primers for influenza, which actually decrease our real-time bioinformatics turnaround times. And now we've got this actually up and running so that as the data comes off of a MinION machine, within 10 minutes, 20 minutes, you can actually start getting real-time data about what's in each one of your samples, including subtyping of HA and all 8 internal genes. So this is going to be absolutely critical, not only for outbreak response, but also for surveillance in remote locations. This has been critical, as I said, not only for our surveillance, but also outbreak response. Here, you're looking at 2 cases of human H9N2 from Cambodia, one from 2021 and one for 2022. As you can see, the human cases in red were quite close to chickens sampled on the farms of these individuals. We were able to actually go from sample to sequence within 8 hours of receiving them using this index system. We also are working with a bunch of collaborators, as you can see on the bottom, to understand whether these environmental samples can be combined with metagenomics for even more rapid information. So we're using a number of different techniques. I'm going to show you one that's using a viral amplification panel from Twist that we're doing in collaboration with EcoHealth Alliance under the CREED network. And so basically, if you look in a poultry cloacal swab, this is a pool of 5 swabs. Again, by PCR, this is an influenza positive sample. So again, the majority of what we're sequencing out of it is influenza, but you do see small amounts of other viruses. But with just one single air sample, again, you are seeing influenza, but you can see a lot of other potentially interesting viruses, both of agricultural and human concern, but also a number of other viruses that could be investigated within that environment. And that could greatly increase your surveillance program over time. So in terms of the future, I think it's absolutely critical to maintain longitudinal surveillance wherever you are in wild animals, domestic animals, and humans. It's absolutely critical we have everyone buy into these systems and understand that they're expensive, but they're necessary. We need to continue to improve that detection and testing pipeline as well. Really to detect, endemic subtypes, novel incursions, what routes of introduction are happening, and evolution of those pathogens are happening. We need more studies at the wild, animal, domestic, and environmental interface. These are high-risk interfaces, not only here in Southeast Asia, but also in Europe and in Americas, and these are important that we continue and coordinate. We need to incorporate and test more novel technologies. There's a lot of good technologies out there, including from INDICAL, that will help us to do this surveillance in more cost-effective and efficient manner. And this is critical for early warning over time. We need to understand the socioeconomics of these diseases as well. So as Jean-Luc showed you and other people have shown, the poultry value chains and movement of poultry and even impacts of COVID-19 are important to understand for behaviors that might affect prevalence, risk, and reassortment of these pathogens. And can we actually implement new interventions for this? What Jean-Luc alluded to at the end here too is also strategies for response and control. I didn't have a good video here, so I just put a pink chicken I saw here in Cambodia the other day. Really, understanding stamping out, protocols of stamping out versus vaccination, the changes in surveillance that will have to happen alongside incorporation of vaccination strategies. And I reflect what Jean-Luc said is that, there's a real need for global communication and collaboration on not only vaccination strategies, but also for choosing vaccination strains and development of new vaccines that we can use, and really looking at the stigma of vaccination and how that might impact reassortment and other things versus the economic impacts of stamping out, especially when things are affecting our flocks so greatly at this point. And then, of course, surveillance for other diseases. When we're out doing surveillance, it's important that we use these samples not only to look for certain diseases, but to look for other pathogens as well. And with that, I'll go ahead and thank everyone that works with me and our funders. I've probably forgotten some people here, and of course INDICAL for inviting me to do this talk, and such a good public-private partnership that we have working with all of their reagents and tools. My email is here as well in case anyone would like to contact me directly. Thank you very much.
Thank you, Erik. Great presentation. I want to thank both Erik and Jean-Luc for these insights today. And we're going to begin answering some questions that were submitted before today's webinar in the registration. Our first question is testing effective in preventing the spread of avian influenza?
Well, that's a big question. I think testing is absolutely critical, not only for detecting what's going on during an outbreak, but understanding what is prevalent in that environment before it becomes an outbreak. And that's what I think I tried to emphasize during my talk, is that if we have an early warning system, we potentially can get ahead of outbreaks before they spread into larger outbreaks, which maybe Jean-Luc wants to comment more on, in terms of if we detect it in a farm, so testing in a farm, if we can contain it to a farm, we can prevent it from spreading to other farms around, or even other countries.
Yes, absolutely. I would only add the fact that the key objective is to avoid, I would say, the start of the episodic. I mean, if you have an index case and you are able to detect as soon as possible, I think that testing is critically important. Since you have spread of the episodics with outbreaks and outbreaks and outbreaks, it will be much more difficult, to be honest, because you will not be able to block the diffusion of the infection. And we do see this right now in some regions in France. Unfortunately, since we have the development in the region, it will be very difficult to block the diffusion. So that means that testing is very important at the very early stages.
Thank you. I think one of the next question from the same person was, what type of testing is most reliable? I think this is a difficult one. So
it depends. I would say it depends on the species, the situation. You have official testing based on swabbing, and we know that it works very well. We know that with this Asiatic lineage, at the early stage, the tracheal swabs or oropharyngeal swabs will be much better than cloacal swabs. But if you consider later stages of infection, it will be rather cloacal swabs. So it's almost impossible to give one simple answer. And it's even more difficult if you have live bird markets and so on. It would depend on the availability, the access to the birds. You have access to the birds. So, Erik?
No, I'll agree with you that it's very difficult to say what one specific thing that you should be testing and with what. Obviously, as Jean-Luc said, we have gold standard tests and things that have been validated and recommended for use. But again, adding on to that, it's not just tracheal or cloacal, it's also going to depend on subtype, where you might detect that more often in tracheal or cloacal, as well as species. And of course, as you choose different sample types, you're going to have different issues with PCR inhibitors and other things that may factor into your testing. So I highly recommend that you follow the guidelines in any given test that you are using, and that you validate those before you implement them under your surveillance program.
Thank you. There's another question here for you, Erik. Are there any updates on zoonotic cases of avian influenza in Southeast Asia? And are there any cases of human-to-human transmission?
Human-to-human transmission, no. I actually think the most recently reported human case was from Spain. Is that correct, Jean-Luc? It was an H5N1 case from Spain.
Yeah, yeah. But we have very few cases, very few cases. And I would.
There still are. No, it's okay. Sorry, it's hard to talk online. Go ahead, please finish.
No, I just wanted to say that it's very difficult to let's say to give a proper interpretation of these cases in humans. At least the last cases notified in humans in United States, UK, or Spain. You have very few cases, and you may have other factors involved in these cases. So that means that we still need to investigate these cases before giving any let's say statement on the zoonotic potential of these viruses. But so far, this zoonotic potential, at least for the 2344B lineage, is very low, very low.
I would agree that there still are sporadic human cases, not only of H5 but of other subtypes here in Asia as well. They're very rare still at the current time, and the zoonotic and transmissible potential of them is still very low. I will say that, however, I'm concerned about the number of mammalian species that we're detecting with these 2344Bs, because obviously every time it spills over into a mammal, there's an increased chance of it becoming more of a problem. So it's something we need to monitor very, very closely.
Okay, we have another question, which is, are there accredited labs in Southeast Asia that offer proficiency testing for avian influenza using hemagglutinin inhibition test method? So Erik, are you aware of anyone that's offering that?
In terms of PT panels for HI, I believe that they're looking at that, especially under the efforts that are now ongoing for a standardized vaccine composition discussion, which may happen on a more regular basis. I know that antigenic testing, so things like by HI, is quite important. There are panels that you can work with reference laboratories in the region, including in Australia and other places, to get standardized sera, but I'm not sure that they actually have a standard PT panel for HIs for avian influenza at the current time.
Okay, thank you. The next question, I think, relates to the AeroCollect, and the question is, are the samples collected can be cultured? So can the samples collected with the AeroCollect be cultured? What types of sample collection control would you recommend? And does the way of collecting the samples need to be the same in different applications? i.e., same height person, same walking speed, same collection time. So Erik, I don't know if you have any input on that. I definitely know that from our experience, we try and standardize it because it's being used as a registered test by DANK and the Danish government. It is collecting the nucleic acid, so the organism isn't viable, so it isn't suitable for culture, but the sample also is essentially ready for nucleic acid testing, whether it be sequencing or PCR directly out of the chamber when eluted in water. But Erik, any other findings from your study at the live bird markets?
Things are relatively nascent at the current time. I'll agree that it's an electrochemical process, so obviously you can't culture out of the sample, but it's very useful to not have to go through an extraction step in between. We try to standardize as much as possible the distance that we're holding the machine and the time that we're using it, but those are ongoing studies at the current time, and I don't have any specific data on them yet.
Okay, thank you. And now, Jean-Luc, our first question from Mohammed Hassin, who's online now, and his question is, have you started in France to vaccinate for highly pathogenic avian influenza?
We started some trials on duck farms, but on experimental basis. It's not implemented at the commercial level, and we need first to validate vaccines, the efficacy, then to obtain the registration of these vaccines at the French or European scale, and to define a strategy. And as I explained, so far we did not define a global strategy, including eventually vaccination based on biosecurity and the surveillance plan. So this is on the way. The trials are going on a good way. There are trials going on in the Netherlands on their use. There are projects of trials in Italy on turkeys, and the idea is to put these results all together to have something like a toolbox with different vaccinal solutions on different species, but to build a strategy at the European scale to be, I would say, to be able to vaccinate and control the diffusion of virus. I stress on this, I'm sorry, but it's very important. We cannot imagine vaccinating without a global strategy with surveillance.
Yes. The next question, which I think could go to both of you, which is again from Mohammed. What about the immune pressure of vaccination in creating new variants of highly pathogenic avian influenza? So I know, Erik, in Asia, they're already using vaccination, and so they probably have a bit more information about these pressures and whether we're seeing changes, but I'll leave it to you two experts.
So that's always been the question is, if we vaccinate and we have a vaccine that performs poorly or isn't quite matched correctly, will it actually result in novel strains emerging over time? I know that there's been a bunch of experiments done, and Jean-Luc may know this literature better than I do, but I know that there's been experiments done in the past where they vaccinated poultry, and again, they didn't see novel variants emerge. And we're not really seeing massive new variants emerge out of populations here in Asia that are associated with vaccine pressure. But the fear is that if we don't standardize and use effective vaccines, that a larger usage of vaccines would result in that occurrence. I mean, just like these viruses will continue to drift and shift, just like human seasonal will be, but it just means that we have to monitor them over time as well. Jean-Luc, did you have more insight on that?
Not really, but I agree, and we do not expect, let's say, some mutations occurring very quickly, and the need of changing the immunoglutinin years after years, such as we observe with the H3 human vaccine. So with the H5, normally we do not expect this, but we will absolutely need to monitor the situation very closely, because we could have novel emergence of a completely different virus, a mix of viruses. So monitoring will be very important, but we do not expect the need of updating the vaccine as we need for human flu, for example.
Thank you. As we're running out of time, what we're going to do is take all the additional questions offline, and when we send out the recordings, what we will do is get our expert panelists to help us with a response for that, because we have a few more questions coming in. But I want to thank Erik and Jean-Luc and everyone today for attending the INDICAL webinar. If you have any other questions, please contact us at experience@indical.com. On behalf of INDICAL and our presenters, thank you for joining us today, and have a great rest of your day. Thank you, everyone.